What if we could actually cut and paste the code of life? Just take a second and imagine having an editing manual for the very genetic instructions that build every single living thing on Earth. I mean, it sounds like pure science fiction, doesn't it? But actually, it's not. It's very real, and it has absolutely changed the world as we know it. Welcome to The Explainer. I am so excited you're here with us today because we're going to unpack the absolute superpower of basic molecular biology that's introduced right here in Chapter 1, an overview of recombinant DNA technology. We're going to break down exactly how scientists learn to rewrite the rules of biology itself. Okay, let's dive into this. Here is our roadmap for today. 1. Rewriting the genetic code. 2. What is recombinant DNA? 3. Meet the biotech pioneers. 4. Medicine, redesigning therapeutics. 5. Agriculture, supercharging our crops. And finally, 6. A century of milestones. Let's get to it. All right, kicking things off with Section 1, rewriting the genetic code, and Section 2, what is recombinant DNA? Let's establish exactly what scientists are manipulating at a molecular level. Now, you really want to notice the emphasis here on direct manipulation. Figuring out that DNA was the ultimate blueprint for life, that was a massive first step. But learning how to actually go in there and directly alter that genetic makeup, that was the real game changer. These methods are collectively what we call recombinant DNA, or rDNA technology, or, just genetic engineering. So how exactly do they do it? Well, the biological toolkit they use is actually fascinating. First, you have to isolate and purify the DNA. But steps 2 and 3, that is where the magic happens. Back in the late 60s and 70s, scientists discovered these specific enzymes that work literally exactly like your elementary school craft supplies. You've got restriction enzymes acting as molecular scissors to cut the DNA perfectly. And then an enzyme called ligase acts as molecular glue to stick different DNA fragments together. Finally, you propagate this newly combined DNA in a host organism. This whole process is what we call gene cloning. Of course, science like this doesn't just happen in a vacuum. So let's transition over to the human story in Section 3, meet the biotech pioneers. And this brilliantly illustrates how crucial collaboration is. On one hand, you had Stanley Cohen. His real superpower was introducing and propagating plasmid DNA inside E. coli bacteria. And then on the other side, you had Herbert Boyer, an expert at using those molecular scissors, the restriction enzymes, to cleave double-stranded DNA so it had perfectly matching sticky single-stranded ends. Separately, they just had pieces of the puzzle, but together, their skills matched up perfectly to create a truly revolutionary partnership. Joining forces in 1973, Herb Boyer and his partner Cohen pulled off something incredible. They successfully cut open a plasmid loop from one bacterial species, inserted a totally foreign gene from another species, and then glued the whole thing back together. No big deal, right? Except it was a huge deal. They created the very first genetically modified organism. Okay, moving to Section 4, medicine, redesigning therapeutics. So they proved they could do this in a lab, but why does this actually matter to you and me? Just think about the stark contrast between the past and present. It's actually kind of wild. Historically, if someone had diabetes, making just a few milligrams of insulin required tons, literally tons of animal pancreatic glands. Treating dwarfism, that took thousands of animal pituitary glands. The supply was insanely limited, the costs were sky high, and, well, because it came from animals, humans often suffered really dangerous immunogenic reactions. But today, thanks to rDNA technology, these exact therapeutics are pumped out efficiently in clean bacterial systems. We have an abundant, super cost-effective supply, and it gives us pure therapeutics with zero animal contaminants. The massive scale of this medical advancement is it's hard to overstate. We're talking about human insulin for diabetes, growth hormone for dwarfism, interferons for cancer treatments, and plasminogen activators, like urokinase, that are used to literally dissolve life-threatening blood clots. rDNA technology is not just some abstract textbook concept. It is literally saving lives every single day. But hey, the power to cut and paste life's code definitely doesn't stop at the pharmacy. Let's look at Section 5, agriculture, supercharging our crops. By employing rDNA technology, scientists can make highly specific, targeted modifications directly into plant genomes. This means we can literally engineer crops to naturally resist devastating diseases. We can build in drought resistance or make them tolerate high salinity soil. For farmers, this is huge. It frees them from the constant, devastating worry of total crop failure when the environment turns hostile. It stabilizes our global food supply, and it even bumps up the nutritional value of the food we actually eat. All right, our final section, 6, a century of milestones. We're going to zoom out now and look at the incredible speed of this scientific snowball effect.
Let's move to the timeline and see how this builds, because it is a total sprint. It kicks off in 1953 when Watson and Crick proposed the double helical structure of DNA. Jump to 1970 and boom, we discover those molecular scissors, the type 2 restriction enzymes. Just 3 years later, in 73, Cohen and Boyer invent actual rDNA technology. By 1982, the FDA approves Humulin, the very first recombinant DNA product. And the momentum just hasn't stopped. Fast forward to 2023, and we're seeing a Nobel Prize awarded for the mRNA COVID-19 vaccines. It is just a breathtaking sprint of human ingenuity.
And the source material for this chapter leaves us with this really incredible quote. I'll just read it. Days are not far when a variety of important therapeutic proteins, peptides, and hormones will be produced from plants employing rDNA technology. Think about the massive advantage there. By using plants instead of animals to grow our medicines, we can avoid viral contaminants entirely. It's going to make our therapeutics even safer and even cheaper. So what does this all mean for us? Well, it brings us to one final huge question. If we can reprogram microscopic bacteria to churn out human medicine, and we can edit our global crops to survive brutal droughts, what part of the natural world are we going to recode next? The foundation we laid out today is quite literally just the beginning. The future of biotechnology is wide open, and I really hope this explainer has left you as fascinated as I am and totally eager to explore the next chapters of this incredible scientific journey.